from the physics background, which should be, the predominant in this meeting, can understand the mechanobiology presentation that is going to follow from Linda Kenney and Mike Sheetz. So just to give a little bit of background, Linda Kenney and Mike Sheetz, Mike, I think, actually coined the term mechanobiology, and they founded the NUS Mechanobiology Institute, which, as far as I'm aware, was the first mechanobiology institute to be founded in 2009. And now, they're back in the USA, but that's been a it's really started a trend, and in a sense it started the discipline of mechanobiology. But when we talk about mechanics, I mean, there is other mechanics. There's classical mechanics, there's quantum mechanics, there's statistical physics, statistical mechanics. And so I just want to, just very quickly give go back to where we started yesterday with this question of what is life. So life well, it's far from equilibrium, as we discussed. And also, as was mentioned, but now will be emphasized much more in the presentation we're about to see, is that it is aqueous. Life needs water, and civilization basically started with development of water management systems, if you like, that could grow food, plants, life, and that could sort of keep us clean and healthy. And in fact, our ancestors in the Indus Valley, they were among the four, early civilizations that developed irrigation, and they were actually the first to develop plumbing architecture around 4000 to 2000 BC. The issue about the fall from equilibrium we have mentioned and discussed yesterday, and you all know, basically this is undergraduate science. The landmarks, the Saudi Carnot and the Carnot circle. So that all happened. I mean, the revolution in thermodynamics, again, it's not, it's more recent. It's about 100 years back, or 150 years back. Saudi Carnot cycle, 1824. The first law of thermodynamics. Von Meyer was one of the first people to articulate that the Joule measurement, the mechanical equivalence of heat and energy, that was all, that's 1841 and 1844. This is my mistake. And so that was the law, the 19th century. And then the 20th century, we get into the actual mechanisms, if you like, of energy conversions in living systems. Otto Warburg was a giant, and we have the fuel molecule ATP and glycolysis in the 1920s, 1924, and then the realization in the 1960s that the way ATP was formed, how you form this fuel, for life energy, if you like, was by electron transfer, either by respiration or photosynthesis, which was then delocalized over the membrane as an electrochemical potential. And that the chemiosmotic hypothesis, again, was one of the main principles of bioenergetics and also of biophysics. Now, the relationship between physics and biology really starts from classical mechanics. So it starts and from imaging, from microscopy. And microbes were first discovered or visualized by Leeuwenhoek in 1673 he made these very fine ground single lenses, which you can see on the right, top right. And that was in 16, and he actually then visualized the first microbes in rainwater. But again, the thesis that there were microbes is very ancient, and in fact, it could be argued that Mahavira, the leader of the Jainism religion, basically he was hypothesized and postulated microbes back in the 5th century BC. And then Ibn Sina in 1020, who lived in Persia and Khwarazm in particular, and had access to the library at Khwarazm, which contained a lot of Indian text and Grecian text, then proposed that these, again, they were not visualized at those times, but those microbes could be infectious and could propagate within a living organism. So the third fundamental property of living organisms is that you have they are organized, and so the microscopy and imaging enable the formulation of the cell theory in 1839. And then the other property is that this organization can be transmitted from generation to generation, Darwin, the origin of species. Now, the mechanobiology and the movie that you're going to show is going to show a lot of motion at the molecular scale. And so the first thing I think to understand that kind of motion is that motion is everywhere. Even inanimate life, when you're small enough, has motion, and that motion is called Brownian motion. And so the ancients, of course, saw because they could only look at large animals, and so movements of large animals, which was directed motion, was taken as one of the fundamental signs of life. But motion at the molecular level exists everywhere. And you have, again, to appreciate that, one needs to know a little bit of history. So the formulation of the atom by Dalton in 1803, then at the beginning of statistical mechanics, the concept of entropy, the Smolachowski diffusion equation, and then, of course, the path-breaking paper of Einstein on Brownian motion, 1905, and then the experimental proof in Perron. So, so that was they basically, that experimental studies of Perron were done on pollen particles, and they proved the Einstein equation, and from that Perron could derive the Avogadro's number. So this is where the statistical mechanics, and if you like, statistical biology becomes starts to entangle. Yeah, and to my mind, it's that era in the last century where biophysics sort of starts, and then biophysics. There's the statistical mechanics, and then there's also quantum mechanics, and quantum biology again has come to the fore, as Sabi talked about yesterday. And I won't go over Sabi's talk. I just wanted to highlight the landmarks again. So the photoelectric effect, of course, again, Einstein, 1905. The term quantum mechanics, coined in 1924. And then another lecture that was influential was the Bohr lecture in 1932, where Bohr actually wondered about the relationship between light and life and atoms. And in that lecture, one of the people attending that lecture was Max Delbruck, who was then important in sort of taking the concept of atomic physics and atomistic particles to then a gene, developing the ideas of the gene, semi-conservative replication, and so on. The involvement of quantum mechanics in biology really has been recognized for a long time. It was first proposed, coherent excitation transfer was first proposed in 1938 by Frank and Teller. And then later in the 60s, quantum tunneling was the pivotal paper there was by DeWalt and Chance, where they were examining purple membranes. These are things which have the light-driven purple pump bacterial rhodopsin, and the temperature dependence of that was quantum tunneling, and they could not explain the temperature dependence by classical methods. And I think that that paper really made people aware that certain phenomena really could not be explained, biological phenomena like a purple membrane pumping, proton pumping in this particular case, could not be explained by classical physics. And then Sabi talked about avian magnetoreception, the triplet and the singlet mixing with the electronic and nuclear spins. And those ideas initially were postulated by Schulten in 1978, but they're continuing to be researched as exemplified by Sabi's work. And where quantum biology has improved, particularly important, has been in bioenergetics and sensory biology. So I have given some papers. People interested in this literature can read up on these references. But in bioenergetics, it's been known for a long time that photosynthetic reaction centers utilize quantum tunneling, electron transfer, exciton decoherence. You know, there's a succession of pigments that then all are recruited to give you the very high energy transfer efficiencies of photosynthesis that approach one. And so there's a big effort in Lawrence Berkeley lab, for example. A lot of money from the Department of Energy goes into this, in making artificial solar cells, which are essentially mimics, if you like, of biological photosynthetic reaction centers. And this is an important alternative to fossil fuels. So right now, about 80% of our current energy consumption comes from fossil fuels. So that can be replaced by, making efficient photosynthetic synthetic devices, if you like. That would be a big gain, both with environmental and climate consequences, as well as for global prosperity. And then in sensory biology, we've two examples, one of which was mentioned yesterday was magnetoreception. And Vidya had this interesting question on the cryptochrome. So I actually looked into that for you, Vidya. So it turns out that there's a very nice paper where, yes, their cryptochromes are involved in circadian rhythms. However, there are multiple cryptochromes, and they're particularly in birds and in fish. And there's a very nice study, and this is a study by Gunther et al., which I would recommend. I kind of found that where this was. They studied robins and compared cryptochrome expression in robins, which are migrating birds, to expression in chickens. And what these people found is that there was one cryptochrome which did not sort of fluctuate, was relatively insensitive to circadian rhythms, but was actually quite sensitive to the seasons. And so, and it was expressed, depending on seasons, and it was expressed in the double cone cells. So these are cone cells, basically cone cells which are linked together by gap junctions to form two cones, and they have been, there's nothing that's been really established, but they have been prime suspects for magnetoreception because it's been thought that their um, architecture, the doublet architecture, could make them sensitive to polarization and other effects. In any case, these Timor biologists showed that the cryptoform 4 is localized specifically in that, and as compared to chicken, which is not the standard home variety chicken that does not migrate, that it also has cryptoform 4, but that is not concentrated in double cone cells. And well, it does. Chickens are, apparently they don't have a lot of double cone cells, and it's not seasonally expressed. So that's one, sort of the biological logic, if you like, that cryptoform. Sami presented the argument from the physics side. This is the argument from the biology side. And then another very interesting paper that Ubaid Siddiqui sort of refereed, our late colleague Ubaid, on behalf of PNAS, is the possible role of vibrational modes uh, in, long-lived vibrational modes in olfaction. So there's this interesting study where you have an odorant, and you can compare hydrogenated versus deuterated odorant, in this case acetophenone. And what the deuterating, it does not change its structure, but it does it adds mass, so it changes vibrational modes. And
Shahid,
it turns out that you could then, this work was done in Drosophila, the model system there. And it turns out that you can actually, if you couple the application of hydrogenated versus deuterated odorant with a punishing stimulus, you could actually then train your flies to, uh, avoid either the hydrogenated or the deuterated odorant, depending on how you paired it. And then they also found some mutants, in fact, that were not sensitive to one versus the other. Atta?
Shahid, sorry, I have to disrupt you. Can you please maybe just wind up because we have like 40 minutes of their talk as well,
Oh, good.
the
All right.
recorded
Okay,
one.
good,
Yes, yeah.
good. All
So,
right. Yeah, no, that's exactly what I wanted you to do. Thank you. So I'm going to just, this is my last thing. So what you're going to hear in the presentation, and I can clarify that on chat for people who have questions, you are going to sort of see images for which you need to have some appreciation of the single molecule imaging resolution. And for that, you first need to understand that, and a lot of people do in physics, but maybe, and also in biology, that lasers have been pivotal. Uh, the physicists would know about laser tweezers, but may not know about uh, the large variety of uses to which laser tweezers. So this is, if you like, the inverse of your refraction phenomena, where instead of bending light, if you have a very strong beam of light, you can trap a small refractile particles, and therefore you can study single molecules. and then techniques for super resolution using photo switchable dyes and so on that give you less, well, basically you beat the Abbey diffraction limit. So you will see in the presentation super resolution images where what you do is that you come along with a very low number of photons and reconstruct the image by not illuminating all of the architecture, but basically dots in sequence and then integrating a movie of images. Electron microscopy, I will not go into, except to say that now with modern detectors and modern microscopes, so this is a microscope on the right where you have the bar is 1 meter, and whereas these are apoferritin molecules, a 1.2 angstrom resolution is now possible in a liquid. This is in frozen vitreous ice. And structural biology is also, a major part of biophysics, which we've not discussed in this meeting. So, Atha, can you maybe start the movie?
Yes, okay, thank you. Thank you, Shahid, for the nice introduction to these.
Yeah, well, hopefully, and then I can elaborate on the,
Yes, sure, sure.
yeah, if there are, but again, put your questions in the chat.
Yes, so, yeah, can you see the video?
I can, yeah.
Yeah, okay, so I'm gonna start it now. Let me just check one thing before so that
Hello, everyone. I'm Linda Kenney, and I'm a founding member of the Mechanobiology Institute at the National University of Singapore, and I'm currently a professor in the Department of Biochemistry at the University of Texas. I want to introduce the topic of how bacteria respond to mechanical stress and present a few interesting examples. Bacteria are sensitive to physical forces, and they experience them as part of their environmental sensing strategies. For example, during growth and elongation, during cell division or cell envelope remodeling, or as they adhere to a surface in order to form biofilms, and as they experience shear stress during colonization and infection as pathogens. Although bacteria have long been known to be sensitive to their mechanical environment, understanding the effects of physical forces on bacterial physiology has been limited by their small size of about 1 micron. But before I begin, I just want to give a shout out to Professor Shahid Khan, first to thank him for the opportunity to give this talk today, but also for his contributions to improving the research environment in Lahore. And several of his students came as summer interns to the Mechanobiology Institute. In particular, Nayab Abidi came to my lab, and Zainab Rahil came to Mike Sheetz's lab in the summer of 2011. And after they graduated, they went on to do PhDs at the University of Illinois in Urbana-Champaign, and they're currently both research scientists at Genentech in San Francisco. And it was a complete pleasure having them in the lab. They were really smart and really motivated. So then let me begin. I mentioned the challenges of studying forces in bacteria because of their small size. It's also a challenge to image bacteria because of their small size. And before the advent of super resolution microscopy, bacterial imaging looked like this, where we have fuzzy images of RNA polymerase here in green and the DNA shown here in red. But once super resolution was developed, we could move to a picture that looks like this from our laboratory, where we image the nucleoid with EDU labeling, and the red is a transcription factor in which we made a fusion protein to photoactivatable M. cherry, And you can see that we can actually count molecules then in response to environmental stresses. And we can also label the bacterial membrane with Nile red. And we can then see how the bacteria change in response to environmental stresses such as osmolality or pH. We can also use our fusion protein for single particle tracking, and in this case, it can tell us about whether or not the protein is immobilized or bound to DNA. So we turn the laser power down and excite only a few molecules at a time, and we can follow those molecules in multiple frames, link up the tracks, determine the diffusion coefficient, which is shown here as a heat map of these tracks. And we're interested in this transcription factor because it's active under acidic conditions when Salmonella is in an acidic compartment inside the host. And you can see that the binding properties, the DNA binding of this molecule increases by 50% in response to acid pH. Now, some fusion proteins are inactive and non-functional, and we can now get around this problem by using genetic code expansion, which was really an experimental tour de force by Kiran Singh, a postdoc in the lab. So we can introduce a stop codon at a gene of interest, and then we use a modified tRNA that recognizes that stop codon and delivers an unnatural amino acid to that site, which can then be specifically labeled, shown here, and uniquely labeled. And it's been really useful for studying proteins that have been difficult to label, or for secreted proteins that don't get secreted when you make large fusion proteins. So then with our imaging, we can really just look and see how bacteria change. So in LB, in rich media, the bacteria that's high osmolality and the bacteria are relatively large. Under acidic conditions and low osmolality, the cells shrink. So we can then image and look at how the number of transcription factors changes and see if their location changes, and we can actually look at the area of the bacteria in response to environmental stress. So we are interested in the signaling processes that are sensing these environmental changes. And in bacteria, instead of tyrosine kinases, we have histidine kinases. And most histidine kinases are membrane proteins. They sense an environmental stress and are phosphorylated on a conserved histidine residue. And then they transfer that phosphoryl group to the second component, which we call a response regulator, that modifies the output. Usually that means binding to genes and activating or repressing transcription. And the system we're interested in is this MZ-on-bar system that regulates the production of outer membrane proteins in response to osmotic stress. We had shown that the MZ histidine kinase could actually function when it was liberated from the membrane and when it lacked its transmembrane domains. And so we were interested in trying to understand what role the transmembrane domains play and what role the lipids play in signaling of this histidine kinase. So we purified and solubilized the MZ protein, and we incorporated it into nanodiscs, which were first developed by Steve Sliger at the University of Illinois in Urbana-Champaign. And then we looked at the activity, and as we went from low to high osmolality, and we could identify changes in the protein through hydrogen deuterium exchange. And what we found is that the ATP binding domain was exquisitely sensitive to the presence of lipids, whether it was after incorporation into nanodiscs, or if we simply took the solubilized MZC protein that lacked the transmembrane domains and simply added lipids to that. So the stimulation of the ATP binding domain stimulated phosphorylation at this conserved histidine residue. And this effect then helps us explain how local anesthetics like procaine can affect MZ activity. So by altering the lipids, this drives an increase in ATP turnover and stimulates phosphorylation. And that mimics the effects that we see of stimulation of MZ at high osmolality. Now, bacteria can exist in the planktonic form when they're freely swimming and using their flagella for motility, but sometimes they can attach to surfaces, interact with one another, and form biofilms. And inside the biofilm, they secrete a curly fimbriae, a beta amyloid fiber, as well as extracellular polysaccharides that essentially wall them off from antibiotics. And so then bacteria can exist and cause disease in this state. So for example, the plaque on our teeth are a bacterial biofilm. And we also see problems with biofilm formation when bacteria can colonize artificial replacements such as knee or hip replacements or heart valves, for example. And here's an EM of a salmonella biofilm that we took. And so you can really barely see the bacteria because they're completely encoded in this polysaccharide and these curly fibers. And this is important because this is a way in which the bacteria can colonize the host and persist in the environment. And one of the important components was the first person who was really made us aware of an asymptomatic carrier was Typhoid Mary. So she was colonized with typhoid, and it's believed that Salmonella typhi forms biofilms on gallstones in the gallbladder. And she never suffered from typhoid fever, but she went on to shed typhi and infect other people in places where she worked as a cook. So a physical interaction with the surface during biofilm formation creates an envelope stress. So here's the bacteria in the planktonic form, and they can interact with surfaces by the flagella or by pili or by the cell envelope. It's not really clear what happens in terms of transduction of this surface stress and how it affects the envelope, but it's maybe through the disruption of an interaction with RCSF and its interaction with outer membrane proteins that then leads to activation of the RCSAB2 component system that then drives biofilm formation. Now, the best example of mechanobiology in bacteria is the way in which mechanosensitive channels are gated in response to membrane tension. So the mechanosensitive channels MSCL and MSCS open and allow for like an escape valve when bacteria respond to a hypoosmotic stress. And this is from a review of Kung, Martinek, and Sukarev, but we have some spectacular structural information from Eduardo Perozzo and others that show that there's an amphipathic helix in the channel that acts to stabilize the closed state. And when the cells shrink in response to hypoosmotic stress, then this helix, which is in the closed state oriented sort of almost horizontal to the plane of the membrane, it realigns, and that leads to an opening of the channels and a release of the higher osmotic contents in response to a hypoosmotic stress. And then lastly, as a great example of mechanobiology in bacteria are these mechanosensitive catch bonds that function during colonization of the host. Now, a catch bond is a non-covalent bond whose dissociation constant increases upon the application of tensile force. So it works like a Chinese finger trap where you pull on it and it gets tighter. Right? And this is advantageous when bacteria are in the intestinal tract or the urinary tract. So they engage with glycoproteins in the epithelial cell membrane of the host through the FIMH protein on the end of these pili. And when they experience fluid stress during flow, then they engage much more tightly, and that enables the bacteria to adhere and colonize the host. Now, lastly, I want to tell you about some of our recent work using Salmonella as an anti-tumor agent. And it's not necessarily mechanobiology, but it's an interesting application of where we can develop bacteria in novel ways to treat human disease. So surgeon William Coley in the late 19th century noticed that some of his cancer patients actually improved when they had suffered a bacterial infection. And he wasn't very good at controlling this process. And soon after that, chemotherapy really came into vogue. And so these results were pretty much ignored. And what we can see here is that when you give a mouse a colon tumor and then later inject them with Salmonella, you can see that the bacteria are everywhere very rapidly. And quite soon after that, they concentrate in the liver and spleen. But then a little while later, they really concentrate on the tumor and they stimulate an immune reaction that then leads to tumor regression. So we are studying this in more detail by using first an in vitro model where we make tumor spheroids and then put them into a microfluidic device containing a hydrogel to support the spheroid. And then we can add bacteria and watch and see what happens, and then use our imaging to actually count bacteria. So this is what an infection looks like, where you see as soon as the bacteria are introduced into the microfluidic chamber, they immediately colonize the tumor spheroid. And we're now using this model to engineer Salmonella to deliver tumor antigens and to study what are the features that enable Salmonella to colonize these tumors and drive tumor regression. And so let me just briefly summarize what I've told you about the bacterial responses to mechano forces. I've shown you that bacteria can respond to changes in membrane tension by gating mechanosensitive channels, by stimulating ATP binding and subsequent phosphorylation of histidine kinases, or by cell body deformation effects on the histidine kinases that drive biofilm formation. Through the use of force-dependent catch bonds, they can strengthen adhesion during infection, and now we're developing Salmonella to study tumor targeting and tumor regression. And so with that, I'll thank you very much for your attention, and I'll now turn you over to my colleague, Michael Sheetz, who's going to tell you about mechanobiology of cancer and of wound healing. Thank you very much.
Shahid, do you want to speak a bit, or should I go to the continue?
Keep going.
Okay, yeah.
So, well, glad to be here, and want to again thank Shahid. Shahid and I had an article together on forces and mechanics of proteins a long time ago. So we've worked together. What I would like to start out talking about is really a broad definition of mechanobiology, and we are really interested in understanding how cells, individual cells of 10 to 20 microns or so, are able to create the forms of organisms in the biosphere. This example of a bird of paradise is a good one because of the ornate and varied nature of the mechanical processes that have to be engaged to create that organism. The issue that I want to talk about today, though, really relates to how repair occurs and wound healing, which leads into the issue of cancer. And how do I get to move the slides
Use your
forward?
cursor and then in the bottom left.
Okay.
And that probably
Oh,
will go
okay,
with it.
fine.
Okay.
Yeah, got it. Thank you. So in the adult organism, you can have remodeling and regeneration. And the extreme example of that is a newt, where you can, after 3, 4, after 9 weeks or so, regenerate a limb. In humans, we have cuts, as here, and you hope that the cells growing back into those cuts take on the form of those that have died. But cancer, it seems, is wound healing or regeneration gone wrong, where if we look at the process, you want to have an adult tissue, and normally adult tissue should not grow. But when you have injury, you want to remove the block to growth so that the cells will grow and replace those that have died. And then you have injury repair, and then you have go back to the normal cycle. Now, in some fraction of the time, it seems that the cells don't go back into injury repair, but continue to grow because they have, you've removed this block to growth, and you get tumors. Many of us have tumors already in our bodies, and most of those are benign. But then when you have enabling adaptations, such as suppressing apoptosis, telomere elongation, so you have multiple cycles of growth, you don't have limited growth, and then you have vascularization. So that leads to a serious tumor. Those cells will metastasize, keep that block to growth off, and you'll have tumors in various parts of the body. But it's really that transformation that we're talking about. And indeed, the issue that was evident early on in studies of cancer is that the cells, when they were transitioning to this growth state, were insensitive to mechanical aspects of the environment, and in particular rigidity. They would grow on soft surfaces, and so we were studying rigidity sensing and found that indeed the tumor cells were missing that rigidity sensing apparatus. So this is the work from Dennis Disher, this summary here, where it was very evident that rigidity is a major factor in differentiation, such that if you take mesenchymal stem cells that are capable of regenerating tissues and you put them in a soft environment, they will go down the pathway to neurons, rigid environment toward bone, and intermediate toward muscle. So, and the chemical aspects of the environment are the same. So it is indeed the physical aspect. And here we see what happens with a fibroblast, which is put down onto a surface, and it spreads out and it responds to the rigidity of that surface. And as you watch it spreading here, you see every 24 seconds, this is sped up, in this middle panel. It is pinching or contracting and measuring the rigidity of that surface, much the way you and I will measure rigidity by pinching an object. And the movement to a constant displacement and measurement of force is indeed a good way to measure rigidity. Now, we talk about the cell as a machine a lot, and indeed you can think about this rigidity sensing process as being performed by a modular machine within the cell. It contains thousands of proteins and can be likened to this automobile engine, and because force, timing, and position all matter and all are critical for that function to be performed. And like the automobile, that modular machine can be present in a variety of different backgrounds, such as the automobile engine can be placed in a number of different car bodies. The rigidity sensor is present in endothelial, epithelial, mesenchymal cells, and indeed performs a similar function in all of those different backgrounds. And indeed, it's on this, what we would call a mesoscale of 220 to 2,000 nanometers in size, up to 2 microns, where it's subcellular, but indeed it's a defined unit of organization within the cell that performs an important function. And we know a lot about the biochemistry over in the left-hand side of single proteins and protein complexes. And we know about cellular activity, but we know relatively little about the modular machines. Now, one of the ways that we have made a lot of progress on this is through the use of nano-fabricated devices. And the one in particular that I'll talk about here is a force measuring device based upon the idea that these pillars, which are made of PDMS rubber, will displace in proportion to the force. They have a spring constant. And we had them made by colleague Jim Hone in the Department of Mechanics at Columbia, and his graduate student went to the nanofabrication facility in Cornell and fabricated pillars of diameters 2, 1.5 micron. Those of 1/2 micron were different than those of 1 and 2 in that they were pulled to a constant distance by cells spreading on them. And that was a feature that we knew about from other studies previously, that as an indicator of rigidity sensing. Same idea I was talking about here, pulling to a constant distance and measuring the force. And so we concentrated on those 1/2 micron pillars, and we had the cells spread on them, and sure enough, when they underwent this cyclic contraction at the edge, we saw that the pillars at the cell edge were pulled inward, and those about 2 microns in from the edge were pulled outward to a constant distance of about 100 nanometers total. There was an actin flow inward, which caused greater displacement at the leading edge than back, but the total was the same at 100 nanometers, irrespective of the rigidity of the system. And you can see that here as we plot the displacement of the pillars. You can see these two moving toward each other. In this case, It was more rigid pillars, so they moved about the same distance. And they did so for about 30 seconds and then relaxed back. When we stained for various proteins that would be in this area, we found that phosphorylated myosin light chain at super resolution here gave evidence of a bipolar myosin filament here. And around the pillars we saw alpha-actinin, which is known to anchor actin and Z lines. So this is essentially a muscle sarcomere that's pulling by the anti-parallel actin being pulled by a bipolar myosin filament. When this was analyzed by Hagai Wolfenson in real detail, so this is using a 100 frame per second camera, and each pillar position was measured in that 10 milliseconds with a precision of about 1 nanometer. And then you average that over 15 frames, and you can get down in the angstrom range. And what you find is that the cell steps, or the pillars are stepped here as they're being contracted, and the steps are about 1.2 nanometers. Now, if we then ask the question, would two pillars that are being pulled toward each other, are they being pulled at the same time? The answer was yes. So the one on the left, which is nearest the cell edge, and orange here, is being displaced at the same time. The green one is being displaced inward, and this is being displaced outward. And that time separation here is a Gaussian around 0, and non-paired pillars don't experience that. The total displacement here of about 2.5 nanometers is an anatomical feature or structural feature of actin filaments. Actin filaments over here on the right. The actin monomer here takes up about 5 nanometers along the axis of the filament. And if you move from this monomer on the front side to that monomer on the back side, you move about 2.5 nanometers. So, and we have a model out in a Nature Physics paper on actually how this is happening. In terms of what is actually sensing rigidity, it seems that the cell moves to that constant force threshold of about 25 piconewtons, and when that is experienced, the cell pauses in its stepping for about 2 seconds. And we see on rigid, it occurs after 4 steps, on soft after about 10 to 12 steps. And so it's roughly in that 20 to 30 nanometer range, 20 to 30 piconewton range, which is what we had measured previously with laser tweezers as an activating force. And when that activating force is experienced, we see here using fluorescent GFP alpha-actinin, we see that the alpha-actinin concentration around the pillar increases dramatically as that pause occurs. So there is a biochemical correlate to this. And we think that is activating now the reinforcement of those adhesions at that site. Now, we've studied this in detail and looked at a variety of proteins that are part of this complex. Many of them are tumor suppressors, such as death-associated protein kinase, the tropomyosin 2.1, PTPN12, and they are all part of this process. On rigid surfaces, you activate growth. On soft surfaces, you activate death through the death-associated protein kinase that's released by PTPN12 with help from the tail and head. So, and as I say, in transformed cells, we have no evidence of this. And indeed, we looked at this in some detail. Does transformed growth here in cancer correlate with depletion of the sensors? The first example we had of this is the MDA MB231, MD Anderson mammalian breast line 231. and it's missing this tropomyosin 2.1. MCF10A is a normal epithelial line, has it. The normal epithelial line doesn't grow on soft agar, but if you remove tropomyosin 2.1 from it, it does grow. So indeed, it's possible to take individual components of this machine, the rigidity sensor, out, and then you don't have it in those cells. And that occurs by depletion. In this case, it occurs by depletion of that gene or by overexpression of a microRNA 21, that's associated with wound healing in many instances. Now, if we add back tropomyosin 2.1 to those MDA MB231 cells, they now pull to the same distance on soft and rigid surfaces. That's this center panel here. And on rigid is blue and soft is red. We see that the parent line pulls more larger displacements, on the soft than on the rigid. And in general, much higher forces are generated in cancer cells. If we look for these contractile units, we see that many more of them now form when you add back that tropomyosin. And when you look at cell growth, you see that the parent line grows on soft agar. When you add back tropomyosin, it doesn't. But we can also play the game of now, when this quote-unquote normal behaving cell, we can remove another component of that rigidity sensor, and this is now myosin 2A. Now it pulls much less. There aren't the rigidity sensors there, but now it grows on soft agar. So it seems that you can now transition the cell from a normal behavior to a transformed behavior by removing or adding back one of the components of this rigidity sensor. And when you do that, you don't change just one protein in the cell, but if you look at RNA-seq, you see almost 1,000 proteins, over 1,000 protein mRNAs are changed, whereas in the human foreskin fibroblast, a normal cell, where we knock down tropomyosin 2.1, nearly 700 mRNA levels are changing by a factor of 2. So it is a concerted state change that happens. Transformed cell state and the normal cell state are different. And we can do that with removing of any one of a number 8 of these proteins that we've validated now will cause cells to become transformed, i.e. to grow on soft surfaces. And I refer you to this annual review Cell Developmental Biology article talking about those 2 states. And what we wanted to know then is if we assume now that many tumor lines are transformed, is there a way that we can selectively target that transformed state to cause tumor death? And interestingly enough, we tried quite by chance to stretch them. And we'd shown previously that stretching of normal cells causes them to grow on soft surfaces. And surprisingly, when we stretch the cancer cells here, the red bar indicates that the apoptosis of those cells was increased dramatically by overnight stretching on soft surfaces, whereas fibroblasts here would die on those soft surfaces unless they were stretched. If we took those cancer cells and added back tropomyosin 2.1, now they started to die on the soft surfaces, but would grow if we stretched them. And conversely, if we knocked down 2.1 and caused the fibroblasts to be transformed now, they would grow on soft surfaces and would die when we stretched. Stretching can't be used as a treatment or therapy, so we looked at another way of mechanically altering cells, and that happened upon ultrasound at a suggestion by one of our colleagues. And lo and behold, if we now use this chick embryo model of tumor growth, the CAM model, where you inject, in this case, MDA, MB231 cells in a matrigel plug below the choriontonic membrane, you see that in these control cells, the tumor grows very nicely. Its green fluorescent GFP is expressed in it. But when you treat with ultrasound for an hour on each successive day, you see that the majority of them are killed. So that process is a calcium-dependent process, unlike the death of normal cells on soft surfaces, and indeed involves an ER mitochondrial stress pathway. So getting back to our model, then if you are transformed, you are under different controls, and you are mechanically sensitive. And so we go back to developing a therapy for this, mechanobiologics. And this is a picture from an article that says exercise inhibits cancer growth. And there was a recent one in 2020 as well, essentially saying the same thing. So mechanical activity, and indeed there's a severe massage therapy that's thought also to inhibit tumor growth. So to give credit here, AJ worked out the ultrasound methods. Guy Wolfenson worked out the initial definition of the rigidity sensor with the help of Giovanni Mecchi. Jim Lohner did the myosin contraction studies. Beverly Lang did the tumor studies. And again, we had collaboration from Shalom Wind and James Hone Mechanic. And I thank you very much. And indeed,
hello, Shahid.
Yes. Okay, so good. So I think we're pretty much, we're still running a little bit behind, but we're mostly caught up.
Yes.
But I do not see any chat questions. So either everybody's understood everything or
yes.
or yeah, because I don't know how else to sort of encourage people to,
Yes. I think
yeah,
maybe, yes, maybe some of them are new to the, like, it's quite new stuff for a lot of them. And
yeah, I mean, I think basically even to ask a question, you need to be at a certain level.
Certain level, yes.
I
And
mean,
what,
if you don't know
yes.
anything, you won't know what to ask. I mean,
Yeah,
so
and also
I think,
the terminologies which are used here also puts people off sometimes, because you use specific terminology which we are not aware of, and then so it's like it was a very like nice, like, two presentations. So the first one I was kind of like asked is, like, in the biofilm environment which Linda was talking about, where the motion is kind of triggered by the osmotic pressure.
yes.
Is the osmotic pressure the only factor determining the dynamics, or whether you have other, like, active sources of motion which contribute to the overall dynamics of bacteria within a
Well, so
biofilm environment?
I think so. She was talking about these mechanosensitive channels that are
Yes.
in the membrane,
Yes.
and so basically a bacterial cell is an osmometer because it has this rigid cell wall on the outside. But then, so if you put it in high salt, the inner cytoplasmic membrane would crinkle up, and so so there's a very significant osmotic force, and you can test that it's osmotic because we know that osmosis is colligative, right? So essentially NaCl would have twice the effect, or than
Yeah.
glucose would have, and things like
Yeah.
that.
Sure. Sure.
So they've done all of that. I think one of the so for the physicists, I mean, for somebody like yourself, I think what this thing is, there are two things. I mean, well, really, in every talk has a jargon. I mean, the calcium signaling session had a lot of jargon, I thought,
Yes.
and in fact, every I mean, every